So I'm really happy to be here today and provide you with an update on our spinal cord injury therapy with OPC1. My name is Francois Binette. I'm the Senior Vice President for Product Development for Lineage Cell Therapeutics. Let me get this out of the way quickly. We're a publicly traded company, so I'll be making forward-looking statement. I'm sure you've seen this in other presentations as well, so I direct you to our website and our 10K filing if you want to learn more about this exciting area. So what is Lineage? We're a regenerative medicine company, obviously. We're manufacturing off-the-shelf cell therapy from our platform of pluripotent stem cell to address severe disease and injury. So those diseases that do not have approved therapies or therapeutic solution right now. So what we're doing is relatively simple. We're taking pluripotent stem cells and we're deriving from these pluripotent stem cell various lineages of various specialized cells. And what we're putting in people to treat their disease or injuries is that we're not actually injecting people with stem cells. We're really trying to replace disease or dead tissue with specialized cells, the cells that have been damaged, and hopefully provide a long-term benefit. So this is really a little bit different than treating symptoms. And this is really what attracted me to this area of regenerative medicine, is potentially the promise of bringing a cure to patients, at least providing a long-term benefit. But that's easier said than done, obviously. To be successful with regenerative medicine, it's rather complex. Not only do we need to understand well the biology of the stem cells, as well as the specialized cells that we are targeting, we also need to consider where these cells are placed in the body. That is, often requires specialized tools and instrumentation that do not exist prior to this therapy being delivered. So we have to work with engineers and develop creative solutions for delivering our cells, because they oftentimes need to be placed in a very specific anatomical compartment. Moreover, we need to have a strong partnership with a surgeon that are enabling with their new surgical procedure to place these cells in the right anatomical location using these new tools and devices. So it's a very exciting field. Obviously, you get to interact with a lot of different people with a lot of different backgrounds, but it's complex. It's different, a little bit different than traditional drug or biologic development. So we have three ongoing programs at Lineage. We have a retinal pigment epithelial cell for macular degeneration. We also have a dendritic cell program for as cancer vaccine, which I'm not going to talk about. Today I'm going to talk about the oligodendrocyte program, or the OPC1 program for spinal cord injury. So spinal cord injury, as you know, it's a very devastating condition. And what I want to show you on this slide is, obviously there's a lot of statistic on that, as far as cost to the patient, to the society in general, and burden on the healthcare system. But what's important, I think, for you to understand is, depending on where the injury is on the spine, it really affects the patient differently. That is, the higher, the closer the injury is to the brain, the more severe or impactful the injury is going to be. So alternatively, if it's lower on the spine or closer to the thoracic spine, it's less impactful, but there are fewer nerves that are involved in this area of the spine. But the problem is that most of the injuries, because of trauma, they occur in the neck region or cervical region, because that's the least protected area of the spine. So this is the area that's more mobile, obviously, as you know, and it's the least protected, because the lower and the spine is protected by the torso and the rest of your body. Needless to say, for a patient, it's really impactful. It's devastating. Oftentimes, patients are wheelchair bound after that, paralyzed, as you know. Lots of pain, recurring pain, chronic pain, rehospitalization. Oftentimes, these patients need to have breathing assist as well, shortened life expectancy. So it's just dreadful for these patients. And it really became very personal quickly for us at Lineage, because although we acquired this program about a year and a half ago, when we merged with a company called Asterias Biotherapeutics that was developing this particular program, Just this year we had a colleague of us, and we're a small company, right? So, colleagues for us are almost family members and friends. So a colleague of us, one of his family member actually got a spinal cord injury, just an accident, and broke his neck. And what's happening is that we got really frustrated in some ways, because although we might have had a therapy that could have benefited this particular patient, we couldn't do anything, because it happened that we finished enrollment in our trial about 2 years ago, for that I'm going to talk about a little bit, and then we're not quite ready for the next stage trial. And I'm going to tell you a little bit about what we're doing in order to get ready for the next stage trial. So frustrating, obviously, for everybody. So it became very, as I said, personal to us as well. So what is OPC1, our therapeutic candidate for spinal cord injury? So OPC1 is a preparation of oligodendrocyte. These cells, what they do, they're not neurons or traditional nerve cell that are part of the brain and the peripheral nervous system. They're the cells that are wrapping the nerves and the axon of these nerves to really insulate these axons to conduct the electrical current or the communication between the brain and the periphery. So not only these cells, they allow or they accelerate this electrical current to communicate between the brain and the periphery, but also responsible to maintain good health of the neuron and the brain and the periphery. So they're very important cells to just maintain and improve the health of brain cells, if you will. So what we found is the cells that we can derive from pluripotent stem cells that we derive into these specialized oligodendrocytes, they do what we were hoping that they would do. We have a lot of preclinical, in vitro and animal studies that show that these cells can indeed remyelinate these axons, that is, they can recoat these axons and the neuron and insulate them, but also involved in general health. They help in tissue remodeling, that is, the help healing of this condition when you have an injury to the spine. They help bring new vasculature to the injury site, and they prevent scar formation. They also help neurons communicate between one another by enhancing these neurite and these fibrils that communicate between the neurons. And more importantly, they improve motor function. So this is what we're trying to do from a clinical perspective, is improve patients' motor function. On this slide, what I want to show you is on this table, you see on the left is a few function or daily activities that people go through for in general life, right? And you have a series of these daily activities, and what's important to see here is that if you have an injury to, say, the cervical level 4, C4, you can see in red here that, if you're impacted at that level, in red meaning that you need total assist to execute all of these normal daily activities, whether it's eating, combing your hair, moving about, in a wheelchair or whatnot. You need total assist for doing that. But if you can heal or improve the motor function by just 2 levels in the neck, remember in one of the first slide I told you that the higher, the more impactful it is. So if you can move this healing down to just a little bit in the neck, you can greatly improve functional recovery in these patients. And now instead of requiring total assist, they can actually be completely independent, and many of these functions require partial assist. So just 2 motor level improvement is really impactful for these patients and their quality of life. And that's what we're trying to attempt with our therapy. So we presented the data on our phase 1/2 trial before. It's been made public. This is the data at 1 year. This was the first time that this product was tested in the cervical spine. And we tested 2 things. We tested 3 different doses because we didn't know which dose was going to be optimal for our treatment. So we tested 2, 10, and 20 million injection. And we tested 2 different of injury severity, right? What's called Asia A and Asia B. And that's not really critical for now, but what I'll tell you is that After 1 year, this was a trial mostly geared to assess the safety of the therapy and gain some, at least preliminary insight whether the product had activity. So we're really happy that after 1 year, we got successful engraftment. That is, the cells were able to stay in the spine and do what they were supposed to do. And we had no adverse event that was created or originated from the product itself. But more importantly, we saw that all of these patients had at least one level of functional recovery, and about a third of the patient had 2 or more functional recovery. So it seemed to be, giving signs of activity that were really promising to us. And quickly after 2 years, we provided that update last year. Continued very good safety profile in all of the patients that received the product. Good engraftment as well, sustained. We can see the cell remain on site for long term, which is what we want to see. But more importantly, in one of the cohort of the patients, so one subgroup of the patient, the group that's mostly similar to what we want to structure the next trial to be, we saw that 5 out of 6 patients actually had 2 levels of functional improvement, and one of them had 3 levels of functional recovery. So really promising. It's not powered to demonstrate efficacy, but really exciting signs of activity for the product, and again, maintain safety. So what do we do moving forward? So when we acquired this program, we knew there were 2 things we needed to do in order to take this to the next level, meaning that to provide material for a large clinical trial and ultimately get FDA approval and start commercializing this product. So we knew that we needed to improve the manufacturing process for making these cells because it was based on a rather old process. And we knew that this process had irregularity in the sense that it was difficult to control the cells from pluripotent state to the specialized state, if you will. And oftentimes, we had impurities in the final product. We need to discard batches of product. And also it was difficult to scale. It was based on small scale, small batches of product, which is not really consistent with, large scale clinical trial and certainly not with commercialization. So I'll tell you a little bit what we did for that. And finally, we knew also that, remember when I told you that we needed a creative engineering solution, we knew that the delivery system, the rig or the device that was required to position very accurately the cells in the spine needed to be improved to make it more user friendly for the surgeon to manipulate this device more easily, as well as just the logistic of assembling this device in the operating room and then managing this device as from the patient as well as from the doctor's perspective. So I'll tell you a little bit about that. So quickly, we did 3 things to improve the process, right? Starting with the pluripotent stem cells, what we did is we generated new cell banks that were much more potent, much healthier, if you will. They could grow and replenish themselves, much better. Secondly, we developed a differentiation process. So the process from deriving from pluripotent to the specialized stem cells is now much better, tightly controlled. It's very easy for us to follow where the cells are going and how efficiently they're transitioning from non-specialized to specialized. So much better control with better growth factors and in-process control. And at the end, we ended up with a much purer product that hopefully will be much more reproducible and that we can actually scale to commercial scale. And so right now, we've done all of these things successfully. We're in the process of now making these cells under GMP, that is, to generate material that we can actually put in patient in a clinical trial. The way to do that is we need to talk to FDA. As you know, we're a regulated industry. So we're talking to FDA about our plan and how we want to introduce this process into our next clinical trial. Similarly, for the delivery system, I've told you a little bit about some of the limitation of the initial delivery device that we had. We tested newer prototypes now that are much easier to use and much easier to set up, to sterilize and to distribute to the various sites. So we tested a few, we did some feasibility testing, and likewise, Our process right now is we're trying to discuss with FDA about some of these changes and how we're going to introduce them to the next clinical stage so that we can do this safely and hopefully we can get efficacy reading from our next trial. In conclusion, from a development perspective, what we need to do now, we need to make clinical grade material under GMP such that we can take this product and take it to the patient. We need to qualify a new delivery device. That is, we need to prove that it's compatible with our cells and the surgeon at the end can use this device effectively. The process now is to work with FDA and have a conversation with them, explain to them what we want to do, and get their buy-in. So we're hopeful that in the next few months, we'll be able to move a much better product as well as a much better delivery system to really facilitate the overall experience from the surgeon perspective all the way down to the patient with a much more robust product that hopefully will provide a functional benefit to the patient. Thank you.